Here is a review of what we just saw in the video. On the left is prokaryotic replication. Remember that prokaryotes have one circular chromosome, and that replication proceeds around that chromosome until two new circular chromosomes are produced. One on the right is an example of eukaryotic replication. Each chromosome can have many origins of replication at once to create copies of chromosomes faster. Remember that the bubble expands in both directions as replication continues. Here is a reminder of how bases are added to the new strand. DNA polymerase III will add bases in the 5' to 3' direction on the new strand. The way to remember this is that DNA polymerase III moves towards the 3. Let's review some of the players in the leading strand. Remember, the leading strand can be on both sides of the replication bubble as it extends. Helicase is the protein that unwinds the DNA and extends the origin of replication. The single-stranded binding proteins will help keep the DNA open until replication on both strands is complete. Once the DNA is opened, DNA polymerase III will add new bases in the 5' to 3' direction on the new strand. Now let's review the lagging strand of replication. Because the lagging strand is opened in the opposite direction, this means that DNA polymerase III can't run continuously. It all starts with the primase enzyme that will add RNA primer to the parent strand. This serves as a handhold that DNA polymerase III can latch onto to start the replication process. DNA polymerase III will run until it reaches the previously created strand. These segments are known as Okazaki fragments. RNA primer is replaced by the DNA polymerase I, and then the segments are glued together with ligase. This process takes longer because it is made in segments and must restart at the replication fork each time helicase unwinds more DNA. Now let's review this process again with a video.